So, before we get into the questions, just again, a few acknowledgements. Just to thank McGill University for considering John Abbott College as a site for hosting this Lister series of talks, and of course, the Lister family for funding this wonderful initiative. I'm hoping that there's some college students here who, if they're thinking they're going to McGill, that maybe they get involved in this particular initiative, and we'll see you here in a few years. Big thanks to Andrew and Ingrid who did the reach out and have organized a huge chunk of this, and of course to Karim for helping facilitate a lot of what's happening here today and for bringing lots of cookies. And thank you to our speakers, because it's not easy always to be talking in front of peers about the things that you do, but you guys are doing some really interesting stuff. And part of being a scientist is doing a lot of this kind of public outreach and getting what you're doing out there. Okay. So I'm going to throw it to the room first. If you guys, anybody has any questions, if you want to ask a speaker specific to the research, tangential to the research, by all means. Oh,
No.
are you sure?
Yeah.
You totally can. Yeah, there we go.
I don't
Yeah, you want to, yeah.
I'm just wondering, what's so special about copper ions that lets them kill bacteria that other ions can't?
Okay. Go ahead, Gillian.
So there's two forms of copper ions, so not to get too into the chemistry, but copper 2 ions don't do anything, but copper 1 ions are so reactive. They have just that one electron in the valence shell, and they just react. And that's what makes them so dangerous. And, I mean, there's other antimicrobial metals like silver, but copper is pretty common in water systems.
Thanks, Benjamin. All right, let's go over here. Did I get it right? Ben, right?
Hi, it's about copper. I heard in the pandemic there was a mask with microfibers of copper in it. Do you think it would help kill COVID or prevent the contraction of the disease?
Were you able to catch a question, Gillian?
Yeah, I was. So I'm not a virologist. I'm a microbiologist, which is a bit different. But yeah, so copper can kill viruses, and I did hear about that mask, and yes, it would be effective. There's also a lot of pushes in hospitals to install, like, on door handles to use copper instead of like a more inert metal like steel. I've also heard of it being installed in like subways around the world to try and like reduce the spread of disease. It's pretty cool, honestly. Copper is very exciting. I didn't expect that when I started my PhD, but yeah.
I can share too that I don't know what's in the paint, but the paint and the railing in the A&E building, the orange staircase, apparently it has some sort of antimicrobial agent that's embedded in it. Now, I'll be truthful, it hasn't been painted again since 2012, so I don't know how much of that has eroded in terms of its effectiveness, but that is my understanding of the railing. Did you hear that too, Karim?
Yep.
I think so. Yeah.
It was part of the design.
Yeah. Okay, let's keep the questions going. Anybody else? No need to be shy. No bad questions. There we go.
Well, first off, thank you for your presentation. My question was, how come copper ions can kill bacteria but not like human cells?
They do kill human cells.
So how do we make sure that people don't?
We don't use, normally we're not in touch with that much copper. Like, it would take a lot to kill. So I work with really small concentrations with really small cells. But so for humans, it would take a lot more copper to like kill a human. That's not a thing. But yeah, it can kill human cells.
Thank you.
Questions so far? Anybody else? Oh, there we go. Julius is making one. Almost steps. There you go.
Do we know what it is about biological hosts that makes a cell go from the VBNC state to a culturable state?
So, we're not quite sure yet. Like, it would definitely be because of the other things I said earlier. So, like, because inside of the egg it'd be warm, so it would reverse the stress that was initially put on it. But also, there's something about the embryo itself. We think it might be, as it develops, it breaks down some proteins into amino acids, and so some of those amino acids might contribute to the resuscitation, but that's actually what my colleague is studying to try and find exactly what it is to pinpoint it.
For the study about bitter taste, how would you take in consideration the culture? For example, if you grew up eating more bitter food as a kid, you may like it more even though you don't have the modification of the gene.
That's a good question. Did you catch it?
Yeah.
Perfect.
That's a very good question. Definitely the culture, the genetics, the environment, the education, and also the social classes, also all of those factors will affect the outcome. But the thing about GWAS is that it takes just the genetics into consideration because we often think that the genetics will not change through the whole life. So whatever the effects of the genetic have will have partially on the variance of the outcome, but on the meantime, that all the factors you just mentioned will also affect the outcome. Yeah, but the genetic component is one of the factors.
Any other questions? Yeah, there we go. That's disappointing. No. Actually, I'm going to ask you guys all the questions. I'll start with Maria, just because she hasn't spoken yet. So something I took note of as everyone was talking is that all of your research areas might have a particular focus and you're bringing a particular expertise to it. Like my sense is you're all biologists, I'm pretty sure of that. But everything has a context that's very multidisciplinary, right? So whether you're dealing with the Legionella problem and you're looking at infrastructure, right, which is nowhere near the realm or sphere of a biologist's interests, or copper pipes necessarily, or even if we're looking at taste, like you're bringing in sort of a statistical model to actually explain biological phenomena. How important do you feel it is for you as a scientist who's specializing in a particular field to still have a diverse background, and when do you know what to recruit from that diverse background to help solve your particular problems?
Okay. For me, because we are using genomics for this study, find out the source of infection and study the isolates. So I think bioinformatics is an important thing. But when I started, I did not have any experience in this. But then I started to learn and I got. And I think your question is how much it's important to be into interdisciplinary when you are doing research.
I guess more the recognition that maybe you need to have a diverse knowledge base. So I'm thinking of like the college students that are here, like they're getting a broad experience. It's like freshman year, right? For those of you guys who are freshmen. So you're getting your biology, chemistry, your calculus class. Sometimes you sit there as a biologist and think, when the hell am I going to use calculus? Why am I doing this? And vice versa. You know, why am I doing biology if I want to be a structural engineer? So that's what I'm asking about.
I think for today's research, either you are in biology or chemistry, you need all of the disciplines. Like you need calculus, you need statistics, and nowadays computer knowledge is very important for doing your stuff. So I think it's a good, if you are taking different subjects courses, it will be good, and you can go in any discipline. Yeah.
Maybe I'll throw it to Gillian. I don't know if you guys want to all maybe address the question in your own unique way. Sure. Yeah.
Yeah, well, I mean, you guys just asked me 3 chemistry questions, and I'm a biologist, so I think that kind of answers your question. But outside of research, I studied One Health, which is where you, which is like looking at these health problems with a more holistic view. So you take in environmental factors and social factors, and that's pretty important for Legionella because I'm talking a lot about infrastructure. So like, where is the infrastructure that's maybe aging and might have more Legionella in it, where it's more of a problem, and of course, Legionella affects older people, so that's another social factor that I need to consider. So yeah, you can't just be a microbiologist, you know, you gotta, and I can't just sit in my lab all day studying Legionella, although I would like to do that. It's also about us addressing these other factors and other things that are really important to your research.
I agree with what they said, because it reminds me just like when we are doing a PhD candidate exam. So that's a very long exam for those of you who don't know. It takes about 4 hours of questions, so you've got many questions from different aspects of researchers. Now, some of them we never thought about that. But that exam officer said like it's not only exams how wide your how deep your knowledge in that specific area, but also about how wide your knowledge is, because I think things are kind of connected in this world and nothing just stands alone. And when you are very focusing on very specific point of the research, you might have like this something like maybe also associated with your research. So I think it's always good. It's always good to keep the eyes open and yeah, just pay attention to the other things that are connected.
Okay, yeah. So I find everything more or less comes back to haunt you at some point. And so, yeah, I've done my undergrad and master's in food science, and what's really cool about that subject is that it's super interdisciplinary. So I had some classes on engineering, some on microbiology, a lot of chemistry, and it was just all put together. And you just never know, so I absolutely hated chemistry when I was in high school. And then when I signed up for food science, I just thought, like, okay, I like food, I'll do that. And I didn't realize like how much chemistry would be in it. But then when it's related to something you care about, then the passion can come out in it. And so all this to say, or even for my own project, like in the beginning, I thought I'd only really be focusing on the bacteria, but then I find out that the eggs work to resuscitate it, and then I have to learn everything there is to know about eggs. And so all to say, even if you don't think it's important now, it will probably come up later in your life in some shape or form, but related to your own interests.
All right. Okay. Oh, there we go. Got another question. You can walk to the end.
This is a question to all of you guys. After your studies, are you guys planning to stay in the research business or go in more the industry?
That's a really good question. Arusha, I'm going to go reverse order. I'll start with you this time.
So that's a great question. I just finished my master's, and so now I'm choosing the next steps in my life. And so currently now, like at this point, I've been in school for however so many years. And so now I'm a bit tired of academia. I want to try going into the industry, but then also open to being, maybe the industry isn't that great, so then might end up coming back to academia. But you're forever a learner. And so, right now I'm still trying to figure that out.
Same for me. I'm just wrapping up my PhD studies, and actually we're just talking about this, this Wednesday, something like that. Yeah, me too. I'm kind of like get used to the past 10 years because I'm always stay in the school and get used to this kind of lifestyle. So I'm currently thinking maybe change another type of lifestyle to see. Maybe I don't like that, but I can always come back. So there's never a confirmed answer.
See, the nice thing about a PhD is you get a delay answering the question. So I don't know yet. We'll see where the wind takes me. I'm kind of, listen, if the microbiology lab in Winnipeg offers me a job, I'm there. But yeah, we'll see how it goes, you know?
So yeah, I am open to both sides, like industry and academia. I'm good wherever I can work in the lab and work with microorganisms, so I'm good.
So I'm going to reverse the question. Did you guys always know this is where you were going to end up in this particular scenario right now, working in labs, food science, or otherwise? Or did you just kind of fall into it?
Yeah, I think I knew this because I was always passionate about microorganisms and using bioinformatics. Yeah, so I think I knew this.
I had no idea. It was, so I applied for a master's at my lab, and I applied on a whim. And then Dr. Fauché was like, do you want to do a PhD? I went, I guess, yeah, let's do it. So now I'm here. Very much kind of just happened to me.
I wasn't. I tried a few other things. So I've volunteered in 2 wet labs during my undergrad studies. So I've tried working, playing with proteins that you, very small particles, like you just wait for 3 days, wait for the result, and then, oh, nothing there. So you don't know what happened to our proteins, and you don't see it. And I also try with the plants. Not very liking with the soil or the seeds, and then right now I'm working with the we call dry lab, so I don't actually working with extracting data. We already have the data. We just need to analyze with the statistics, something like that. So I feel like I'm more liking this type of working style because it takes less time to get a thing done, and also I feel like I waste less time for the failure because for the protein example, 3 days nothing. That my failure cost me 3 days. But for now, also 3 days maybe for the model to get running, but I can also get other things at the meantime. So I think that works better for me. But I would say try everything, and then you won't even know what's your like or dislike until you try.
So my story is very similar to Gillian's. In my undergrad, I just needed something to shoot for because if I wasn't aiming for the best, then I'd end up failing the class. And so I was like, okay, my goal will just be to get into a master's program. Kind of be, I might get in, I might get not, but either way we'll try. And so then I ended up getting in, and I'm like, okay, well I'm here now, I'll just do a master's, and that's how it went.
Okay, so I'll leave you guys with one last question, okay? At some point in everybody's talk today, I don't know if you guys in attendance noticed, but you all got excited about something. Something about what you were doing that really sort of your eyes lit up. What is it about science in general that just excites you? And is this a passion for you moving forward? And maybe why?
Okay, let me just think about that for a
Yeah.
second.
I just fell into it. That's exciting.
Yeah. No, definitely. With science, it's just that, you never quite have the answer, and there's always an opportunity to learn more from it. And it's just cool because then you get to learn more about your natural world and explain why things are the way that they are. And I forgot the second question.
Oh, just I don't know.
Awesome.
So you always get excited about, you all got excited about something. I guess the question is, what is it about doing science that genuinely excites you? Makes it interesting, motivates you?
When things work out finally.
But even in the moments where it doesn't work out, and it's funny because there's a few people here in attendance where we talked about a few weeks ago how things don't work out, where we don't know the answer.
Yeah. That's like normal life, real life, it doesn't work out.
But is there excitement in when it doesn't work out? Why might that be that?
Because you want to know why it doesn't work out. And because you already put so much effort into it and it doesn't work out, that makes you even want to try more. Because why it doesn't work out? That's
I really like puzzles, and science is kind of like that. I mean, obviously there's the, when you discover something new, but also just the day-to-day in the lab, when you're troubleshooting, something doesn't work, and you're trying to figure out why. I actually really enjoy that process, so I really like science.
There we go. Maria, you got the last word?
Yeah, it's like Gillian. I like about science is that I'm learning all the time. Even if my experiment is not working, it makes me frustrated. But then I try to find the answer, and even if I'm not finding the answer, still I'm learning something. So I like this thing about science.
Great. So let's give one more round of applause. If you guys still have any questions, maybe that you're too shy to ask, or you want to ask very particular questions, maybe about their life experiences and get some advice, there's plenty of coffee and drink and cookies to talk those questions over. Again, big thank you to Ingrid, big thank you to Andrew, big thank you to McGill University for arranging all this. A big thank you again to all of you. You did a wonderful job. And I hope that, I think this is probably the last thing we're doing, you and I, Karame, this year. So, next year. All right? Okay, have a great day, everybody.